Okay, last week we did all terminology. Sarah also gave you guys an indication that you could look up a glossary of terms that might also help remember some of the things we talked about last week. But by getting the big words out of the way, we're going to be able to go very quickly through a lot of the different processes that are involved in setting a dose for sterilization with irradiation processing. And that's going to be our focus of today. This is likely to have many more questions or more specific questions than we had in the past, but that's okay. What we can't answer today, we will continue to answer offline afterwards. Keep in mind that this is a very early beginning webinar series, so we may not get into a lot of detail on things, but I want to make sure I introduce to all of you the different types of different approaches to being able to set a dose and some of the things that are very critical to that process. Okay, the dose setting process. What are we going to look at today? We're going to look at what guidance is available to you out there for dose setting, what options that guidance gives you, what tests are part of that process, what you do with all this data you collect to support a sterile claim, how much product you really need to do this testing, and long term, how do we maintain the word sterile on our product? Okay. The big guidance documents in this area are the AMI and ISO documents, most importantly the 11137 series, Part 1 through Part 3. Part 1 is titled The Requirements for Development, Validation, and Routine Control of a Sterilization Process for a Medical Device. The important thing in there is that part tells you in general what your requirements are when you're dealing with a healthcare product that you're going to sterilize with radiation. Part 2 is the one where we're going to spend most of our time today, and that is literally the step-by-step parts of setting a sterilization dose. Part 3 will become much more important to you in the seminar, Part 3 of the series, next week on dose mapping. This is where you'll get your guidance on measurement of dose and distribution of dose throughout a product. In addition to the 11137 series, there's an additional ISO and AMI document that's called TIR 13004. It's very similar to what's in 11137 in one respect. It has different dose setting methods and different dose setting tables. But it's specifically to methods that are called VD max, where it's a specified dose, and there's separate tables for a series of methods that we'll talk about in more detail as we progress through this. Okay, the guidance documents, Part 1 and Part 2, are going to, there's additional to the ones that tell you how to set a dose. There's ones that tell you how to test to get the correct dose. They're the ones that a microbiology lab are going to be more concerned with than, for example, an irradiation processor. You had 11137 for setting the dose, but you have 11737, Part 1 and Part 2, for doing the microbiology work. Part 1 deals with more the population of the microorganisms on your product, or the bioburden, as we discussed last week, and Part 2 deals with sterility testing. The other document that people sometimes forget about, that's probably the one that's going to be most valuable to you in your whole career with sterilization, is TIR 17. And it's titled Compatibility of Materials Subject to Sterilization. It's a really good early resource for all the different modalities for sterilization and how certain basic polymers react to those methods, so that maybe you can avoid some pitfalls of using an incorrect or a less than optimal plastic when you're developing a new product. Okay, dose setting options. Within those standards that I referenced, you will have something called Method 1, Method 2, which has 2 different versions, A and B, and then a series of VD max methods. Each of the VD max methods has a number after it, and that number becomes the minimum sterilization dose for your product if you follow that method successfully. The ones that have the little stars next to them are all the methods that are in the 11137 document. The ones without the stars are in the 13004 document. Okay. We're going to look in detail at that 11137, and we're going to start with the methods that are in it, particularly the Method 1, the Method 2s, and the first and most common VD max methods. all of these methods that we're going to talk about today, with the exception of method 2, are all going to directly be based on bioburden. You're going to hear bioburden referenced over and over again because it is the critical factor in a lot of these. For setting the right dose, for controlling your sterilization process, it's all about the bioburden. Okay. The 13004, this is so that you have it on a separate slide so you know which of the VD-Max methods are in 13004 versus in 11137. Now, what tests are done with each of these methods? All of those methods I had on those slides are approaches you can use to set a minimum sterilization dose. Some are going to work better for one product than another, but all of them are options, and all of them give you the possibility of setting your sterility assurance level to what you need or want your product to have. The goal of dose setting is to document and set a dose range. This is the minimum dose required to sterilize the product and still give it the sterility assurance level required, and sets a maximum dose allowed that you will allow a product to have that will not only sterilize it, but at which you know will result in a product that remains safe and effective for its intended use. So the goal is to have something not just sterile, but something that works well when you're done with it, that will be able to perform the purpose that you intended it to without harm or without loss of its specifications. Required for the minimum dose, we actually talked about some of these tests last week, but we're going to go into much more detail today. To set the minimum dose, you should know what your maximum allowable is, because you want to make sure you pick an approach that will allow you to have a dose range when you're done. You have to know what the bioburden is in most cases. You have to know what the bioburden recovery efficiency is, the bacterial stasis and fungistasis, or BNF, and sterility. I always tell people, and that's why it's in red here, it is highly recommended to not start testing for your minimum dose until you know what your maximum dose can be. Setting a minimum dose on its own does not automatically give you support of a given maximum dose. For example, if you look in literature, the most common dose range you'll see for irradiated products will be 25 to 40 kilogray. There are methods that will give you the ability to claim 25 kilogray gives you your sterility assurance level. By doing that work to establish the 25 does not mean you have automatically established 40 as your maximum. Maximum dose is not based on just the type of product. For example, one catheter made by one company and another catheter made by another don't necessarily have the same maximum dose. They could, but they don't necessarily. You have to document that what will work for your product. Maximum dose is affected by the specific choices of materials, the sources of those materials, the production process used to make and assemble the product, how the product will be used, and what you've promised the product will do when it's sterilized. It's a combination of all the things that your specifications say that makes your product unique for its purpose. When I have a maximum dose plan, what do I test my product? Unlike setting the minimum dose, which will be very clear, there will be step-by-step processes in the standard, how many samples to test, what tests to do. In maximum dose setting, what you have from the standards is a nice quote. It says, establishing the maximum acceptable dose, section 8.1 of 11137-1. The max dose for a product shall be established. One, that line right there says you have to do it. When treated with the maximum acceptable dose, the product shall meet its specified requirements throughout its defined life. That means it has to work, it has to be safe, and it has to last for the time you say it will. And just to emphasize that you have to do this, section 8.3 also says the sterilization dose and maximum acceptable dose shall be specified for the product. Okay. How do I test this? Well, I always tell people start off with thinking about what critical functional requirements does my product have? Do I have appearance limitations? For example, What if I have to shine a light of a particular wavelength through a product? If my product was to discolor or darken, maybe I obstruct that light. Or maybe I have to be able to see something when it goes into solution. So the physical properties also have to be considered, as well as the appearance. How do I know? How do you know before a product's been irradiated, when you're designing it, that it meets the performance specifications you wanted it to? Those are good quality indicators for you to test to after it's been irradiated. You have to make sure that when you're testing, when you're developing a product, when you're developing a design, you're going to establish limits for what you consider acceptable. These become your quality indicators as a result, and they're good things to test to after you've looked at what happens to your product if it gets the maximum allowable dose. Make sure that any testing you do, that the product receives the maximum dose you will allow, not the nominal dose, to assess these characteristics. For example, if your product is going to be processed at 25 to 40, test it at 40 plus dose, not at 25 to 40. If 25 to 40 is your range, the 40 becomes your acceptable max. To achieve a 40 minimum dose on your product, the samples you assess will get more than 40 kilogray, but you can request that the dose delivered be as tight as possible. But always remember, what the minimum delivered to those samples is the maximum you can claim. So if you do, for example, 40 plus or minus 10%, and we deliver 39, your maximum now is 39, not 40. You have to know that everything in the product mix that you tested got at least that 40. Always test worst case, not nominal, so that you know what dose you've achieved and what your maximum allowable is. Look at your certificates of processing for any test samples. The lowest dose delivered is the highest dose you can claim to have data for, since you know that everything in that carton or box got at least that dose. Okay. There's two ways you probably have seen certificates of processing. One version is like this, but the two I'm going to show you have the same information. In one corner, you would have where it was processed, the run number for it. This one is from our Radiation Technology Center. The important thing on it is it'll have your product information, and it'll have off to the right the dose range you requested, the minimum you asked for and the maximum, and right next to it, the delivered minimum and maximum. Those are the numbers that are critical for you knowing what your dose product got, whether it be in the sterilization process or the max dose. The other version of a certificate of processing is more along this line, where those numbers, that minimum specified and minimum delivered, are at the bottom of the page. The information is always going to be there, and that's the document you have that proves what your product was delivered when it was irradiated. Okay, why do I want to know the maximum dose first? Well, what if you do all that microbiology testing we talked about last week and that we're going to talk again about this week? What if you did all of that and established a minimum dose only to find that the maximum your product can tolerate is so close to your minimum that the dose cannot be delivered? If the max is too close to the minimum, there may be no room for processing to occur. That's one of the terminologies we mentioned last week, which was dose uniformity ratio. Look at that maximum versus the minimum. If those numbers are too close together, there may be no room for you to be able to process your product. You may find that your minimum, if you say, for example, use the approach of VD Max 25 and did all your testing and everything passed the way you wanted it to, you now have that you can sterilize your product at a minimum of 25 kilogray. If that's true, what happens if when you do your maximum dose testing later, you find out your product can only tolerate 20 kilogray? Now you've got a reverse dose, which again means you cannot sterilize your product. What if you find out your product doesn't tolerate irradiation at all? You will have wasted all of the testing you did up front. Best to know that for sure you can do the process and have some idea of just how high a dose you can go. The other reason this is important is it's also going to help you select the best method for setting that minimum dose, because it'll eliminate ones that you can't do because of the tolerance of the product. Dose setting minimums. What options do I specifically have for my product? Not how many do I have available in the standards. Well, the first place you start is what sterility assurance level do I need, and what maximum dose do I allow? Because those two questions are going to eliminate a lot of the long numbers of choices on that list so that you can get a more manageable set of options and then pick which one works best for you. Assuming you have these two, the choice is going to be much easier for you. Okay, we're going to start looking at all of the key methods based on which ones are that have been around longer, and then we're going to talk about comparisons of them so that you can better select why one might want to use one or the other. We're going to start off with method 1, mainly because this is probably the one that has the most history. It's been around the longest, it's been in many revisions of the standards, but it is always out there and it's still a viable option. In many cases, the reason it is still used is it can set a sterilization dose lower than, for example, VD Max 25, which is very popular, and can even match VD Max 15 or go slightly lower if the product's bioburden is very low. It has modified dose tables to clarify the application of bioburden averages even for products that have an average colony-forming unit quantity for their bioburden lower than 1. So there are actually 2 dose tables in 11137-2 that relate to method 1. Table 5 for bioburdens 1 CFU or higher, and table 6 for ones that are a fraction of 1. Bioburden recovery. First test you're probably going to do in the micro series will be this test. This test is the validation of the method that will be used to extract your bioburden from your product, and its purpose is to assure that the method is effective for your product and that you get an accurate measure of that bioburden. Because like I said earlier, it's all about the bioburden. The bioburden is the thing you have to kill in order to make your product sterile. So the more accurate that number is, the more accurate your sterilization dose will be. There are 2 methods for approaching that are in the standards. One's called exhaustive rinse and one's called product inoculation. Exhaustive rinse measures your actual product's bioburden extracted through multiple rinsing, shaking, vortexing, whatever steps are needed to agitate it to make the bioburden come off the product and go into the media. So you're measuring the actual organisms or bugs on your product and quantifying them. It's done through a series of repeated extractions, usually 4 or 5, measuring the amount of recoverable bugs each time. It compares the initial rinse to the total of all the rinses put together. Okay, so you get an idea of what that looks like mathematically. Say you had 5 samples. You extracted each one of them 5 times, and then you compare the total versus the first rinse. And then you take all of the samples you did, and you get an overall average. In this example, the overall average is 79.9%, which would give you a correction factor for your bioburden of 1.25. That would mean whatever my bioburden number was, the raw number I extracted, how many counts I got, I would multiply that number by this correction factor of 1.25. Exhaustive rinse, the considerations one has to be thinking about is that it will not always reach 0. You could do multiple extractions and still be getting bugs. It can be preferred by some people because it actually measures the real bugs on your product, not something that you inoculated on there. It is not always preferred in handling because it can use a lot of media because of these multiple extractions. It also has to be, you have to be aware that it does not always give a measurable value when the natural bioburden is exceptionally low. Repeated rinses of something that already starts out at 0 counts is probably going to give you a result that will be published as UTT, unable to determine, or greater than 99%. So you may not get a measurable number. It's frequently used, though, when samples are very hard to get or are expensive. since in this case, the samples used for an exhaustive rinse can be the exact same ones that are used in your bioburden. You don't have to have a separate set of samples. If a usable value is not achieved and the bioburden is already done, in this case, though, you would have to repeat the bioburden because you wouldn't have a measurable correction factor. Product inoculation. You need to start off with product that has already been sterilized some way. In most cases, since you're doing irradiation, you would probably irradiate the product first. The number of units you actually need to do the test will vary with the labs or with your preference, but it's usually going to be no less than 3 and usually no more than 5. The goal with the pre-treatment, that irradiation process or other sterilization, is to kill as many viable organisms as possible, because you're going to inoculate that product with a known organism and you want to be able to see it and be able to count it without having it covered over by natural bioburden growth. The most commonly used organism is called Bacillus atrophius, and you're usually going to inoculate the product with less than 100 CFUs, usually somewhere between 10 and 100. You want to have enough that you can see them extracted, but not so many that they're difficult to grow. After inoculation, you do the process of bioburden extraction the way you plan to do it for your regular product. And now you know how many organisms were on there to begin with, live ones, viable organisms. And now you have a number for how many you extracted off. So it's the same thing as in the previous recovery, where you're comparing a known to an unknown. Okay, an example of that, for example, say you had 5 samples and you inoculated them with 100 CFUs of Bacillus atrophius. You measure how many you get off using your preferred bioburden test method. In this example, you end up with 73% extraction, or a correction factor as calculated on the side. Okay. Inoculated concerns. The bugs are placed on the product. They're not the ones that naturally occur, and that it uses extra product samples, usually that are irradiated first. The advantages, though, is it uses less media. It will always get a measurable number, even with very low natural bioburden, and can easily be done before the bioburden is even started. This avoids the possibility of having to repeat a bioburden test because your extraction method was not good. Better to lose 3 or 5 samples than to lose 30 samples for bioburden. You can evaluate that bioburden method up front to make sure you get a good one without subjecting any loss of your bioburden samples. Okay. Recovery in general, its purpose is to improve the accuracy of bioburden data, and as a result, assure that your verification dose, that sublethal dose used for sterility testing in the future, is more likely to pass. If you underestimated your bioburden, you will be underestimating your verification dose and making it a harder test to pass your sterility. The best method is what works best for your product and gives the most reproducible results, as well as a good percentage recovery. Keep in mind, laboratories have a lot of experience in this and are excellent sources and recommendations for the better method. They're also going to have seen many different kinds of products, some that are very similar to your product, and may have good ideas on what methods for extraction will work best. Method 1, what tests do I do? You started off with that recovery efficiency. You're going to now do bioburden. Bioburden, you're going to test 10 samples from 3 different production lots that are in process control, meaning you've figured out how you're going to make your product, you've developed a stable, reproducible process for producing it. The product samples coming off that, whether it be lot 1, 2, 3, or lot 3,000, 4,000, or 5,000, should be in process control. They should be very consistent. You are going to use unirradiated product for bioburden because we want to be able to measure the natural bioburden, how many viable organisms are on there that we have to kill with our sterilization process. We mean final product in its final package because each step of the production process has the potential to add organisms to your product. Up until the point we seal the sterile barrier shut. Bioburden measures how many viable organisms in colony-forming units you have as a result of the product being made in its normal manufacturing process. There's your schematic from last week, just as a refresher, and so we can get a little color in the slides. You've got the product. You're going to put it in some kind of media, treat it in some way, shake it, blend it, and then extract that off onto plates of some sort so that you can incubate them and count how many colony-forming units are produced. Okay. Method 1, bioburden will be determined by the microbiology lab. They're going to measure it in colony-forming units. So when you get your report, it'll have a number, and that will refer to how many colony-forming units they saw. In Method 1, most people will say that bioburden recovery is recommended to avoid underestimating the total bioburden. What it actually says is that you don't have to do it, but it's recommended. But it also points out that if you don't do it, you are likely underestimating your bioburden. So the other thing is, if you don't do it and somebody reviews the data, they're going to ask you why you didn't do it. In the old days, many times people did not do recovery efficiency with Method 1. In the last 10 years, I haven't seen anybody not do it. So don't think of the terminology as a way of avoiding a test. Think of the previous test as one that really is to your benefit to do. Bioburden is used to determine directly the verification dose. In Method 1, the verification dose will have a sterility assurance level of 10 to the minus 2. You're going to be testing 100 samples, and that 10 to the minus 2 is the sterility assurance level on those samples only that you're trying to show, not the sterility assurance level of your final product. The average of the 3 lots tested will be used to set this dose from Table 5 or Table 6 in 11137-2. Monitoring bioburden accurately over time will also be used to evaluate if your production process stays in control. It can alert you to potential changes in your production process. It will be part of what we defined last week as dose audits, those ongoing required testings to substantiate that your dose is still effective. Bioburden is not a pass-fail test, but if not measured accurately, your sterility test has the potential to fail. So it is a critical number and value to know and control in your production process. A piece of what one of those tables look like for Method 1, that Table 5, you would have on one side your average bioburden that you measured through that extraction process, and the first column to the right is a column that's labeled 10 to the minus 2. For Method 1, that is your verification dose. The nice thing about Method 1 tables is they also give you the sterility assurance level for 10 to the minus 3, 4, 5, and 6. The most common of which will be that 10 to the minus 6 column, which is the one that will most commonly be used as your minimum sterilization dose if you pass your sterility test. So in this one table for Method 1, you get both a verification dose and a minimum sterilization dose based on your bioburden. Okay, what do I do with all this bioburden data that you're going to make me collect? Bioburden plus recovery give you that adjusted value for the bioburden, so you can set your verification dose. Your dose of irradiation predicted to give you either a sterility assurance level of 10 to the minus 1 when we start talking about VD max methods, or 10 to the minus 2 when we're talking about either Method 1 or Method 2. Is important because that is your pass-fail test, that sterility test. So the accuracy of that dose is important. This dose is considered sublethal and is used only for testing purposes to establish or verify what the minimum sterilization dose will be. It is not the minimum sterilization dose. For example, you could very easily have a verification dose of like 5 kilogray and have a minimum sterilization dose of 25 kilogray. You're testing at lower sterility assurance levels to document that at the higher dose, you have the sterility assurance level you're required to have. Product cannot be sold or used labeled sterile if it gets only the verification dose. It is selected based on the dose setting method and will not be the same with different methods, even if the bioburden number is the same. There are different dose tables for each of the methods we're going to talk about today. We did this so far. We've talked about the bioburden half of testing. You have a test to assure the accuracy of the bioburden in the recovery, and then we have an accurate number that we get off of the product through bioburden extraction that we use to set the verification dose. The next half of the testing is all related to sterility testing. Just like recovery efficiency, you had a method for establishing a valid method for doing sterility. You had recovery for bioburden. Now you have something called BNF testing, or bacterial stasis and fungistasis, to establish an appropriate method for the sterility test. So you're validating the method. You're assuring that the test conditions when you incubate for a sterility test will allow an organism that is present to grow. Not just keep them alive, but allow them to actively grow. An organism present that does not grow cannot be seen in a lab. They can't tell that the product maybe had living organisms in it unless they proliferate. The end result is if you don't have an effective BNF test, a passing BNF test, your product could have a false negative in a sterility test. So you may have more confidence in your results than you really deserve to have, because you could have organisms present that are alive and that when they're put in a different condition will grow, but in your test they did not. BNF testing on most devices is done with 3 organisms, specifically a mold, a yeast, and a bacteria. There is the possibility of doing 6 organisms or 6 tests, where most commonly with pharmaceuticals or products where certain organisms may be present. In that case, you'd have 3 different organisms for 2 different media types. And what you have here in the picture is the ones that look kind of cloudy off to the right are the ones that are growing. And on the left, the ones that are clear are the ones that are not growing. And this is the one test where you really want the bugs to grow. You want to know that under these test conditions that a living bug will grow, because you want to be able to see it if it's present in your product. Okay. Test is media specific and volume specific. A test done in soybean casing digest broth, which is common for aerobic organisms, that is done, for example, in 100 mils of media, is not a substitute for 100 mils done in fluid thioglycolate media. The FTM media is used more for anaerobic organisms and has a different incubation temperature. The 2 medias are meant to encourage or support the growth of different types of organisms that may be present. A passing test at 500 mils of media is not equivalent to the same test done with only 100 mils of media. In that case, the 100 mils with the same presence of product could have a more concentrated presence of material. So it could be more inhibitory than the more dilute 500. But turn that around, if you had a passing BNF test with 100 mils of media and then do the test in 500 because somebody was just generous that day, that test is still good because all it did was provide more growth media. But if anything, it diluted out any negative effect from the product's presence. You're using this test to determine if microbiocidal or microbiostatic, keeping things from growing versus killing them, agents are present in your product, and that they get extracted into the growth media and inhibit growth of organisms that you want to know if they're present. It is important, especially when you're dealing with a new product in a lab, that if you know anything about the product having an antibacterial property to it, meaning it has an antibiotic on it or it has some natural material on it that's known to be antibacterial, that you let the lab know before they test it. In many cases, they can take that information and take it into account when developing the test, or find a way to neutralize it and save you the cost and time of having to repeat a test to try to get an optimal result. If they know upfront what could be a potential cause of a problem, they can address it right from the start. BNF is an inoculation test, just like that inoculated recovery test. In recovery, you used one organism and you just extract it. In BNF, you're using 3 different kinds of organisms. The most common ones are Bacillus subtilis to represent the bacteria, Aspergillus for mold, and Candidus albicans for yeast. Some specific labs add a 4th, a particular one, Micrococcus luteus. The goal of a BNF test is if I put 100 CFUs of these organisms or less on your product, I have to be able to recover what I put on in there within 5 days of the inoculation. Okay. Remember, with BNF, you want the bugs to grow. So if they grow, the test is passing. Whereas in a sterility test, a bug that grows is a failing test. Okay. That should be sterility on top of that slide, so I apologize for the title being missing. The conditions for the test of sterility are based on the results of your BNF test. This is one of the easiest tests you're going to do. You literally, a lab is going to take your product after it's been irradiated, put it in media, and incubate it for 14 days. It's a yes-no test. Did something grow or did nothing grow? Not how much grew or what grew. Any growth in a sterility test is a positive for a sterility test, and you have to count up how many of the incubated samples had any growth in them. For method 1, this test is done with 100 samples, all from one lot. There's your summary from last week of what a sterility test is. You have the samples irradiated at the verification dose. They're put into sterile media, and then they're incubated and scored at the end of 14 days of whether they're cloudy, meaning something grew, or they're not cloudy, which means nothing grew. Okay. All those tests you did for method 1, we're going to do the same tests with all those methods that are labeled VD max. There will be some differences, but you still have to have a max dose for a product that's validated VD max. You still have to do bioburden. You still have to do a recovery efficiency. You still have to do BNF, and you still have to do sterility. The difference and the reason why many people prefer a VD max method is you will use less samples, which means you'll have less cost because each sample you test has an additional cost. And if your product is precious to you, you may not have very many samples to be able to do 100 for a sterility test in method 1. What makes VD max methods different from method 1? Bioburden is still 30. Recovery is still done the same way. BNF is the same. Your big difference is the sterility test. In a VD max method, when you get to the sterility part, you are testing only 10 samples from one lot, not 100. All of the VD max methods support a sterility assurance level of 10 to the minus 6. The dose tables don't have on them those other SALs that you saw on the method 1 table, so they don't have the column for 10 to the minus 3, 4, and 5. The verification dose is a sterility assurance level of 10 to the minus 1. That relates to the fact that you're only testing 10 samples. There are multiple dose tables that are titled VD max. You have to make sure you select your minimum dose, the minimum dose when you're selecting your doses for your verification dose. If you're doing a VD max method, you have to make sure you're looking at the right dose table to match the method you're trying to do. Because for the same bioburden looking on different tables, you will get a different verification dose. Okay? All these VD max methods are available. Each one, the number reflects what the minimum dose for sterilization will be to achieve a sterility assurance level of 10 to the minus 6 in your terminal sterilization process. But because of those numbers, they have limits on what your bioburden can be and still apply that method. For example, you might say, well, I'd like to have VD max 15 because I want my minimum sterilization dose to be 15. But what if your bioburden is something like 200? VD max 15 has a limit of 1.5 CFUs on the bioburden, so you would not be able to do VD max 15. You would then, if your bioburden was 200, you could potentially do VD max 22.5 or one of the higher bioburdens. All of these methods, if you're within the bioburden range that allows you to do them, all of them will support a 10 to the minus 6 sterility assurance level. Here's a little selection from the VD max 25 dose table. It will look very different from that piece I showed you of the method 1 table. And that's because you have average bioburden and you have your verification dose. But you have something called a SIP reduction factor, which is only used in calculation when you have bioburdens or when you test a product where you don't test the entire product. One of the other definitions we gave you last time was SIP, sample item portion. And a SIP of 1 means you test the entire product, in which case your verification dose will be that first column, or second column, the one in the middle. And if you had to use a fraction of a product, say you were trying to test an entire drape, and you couldn't get the entire drape in a container, you might test a fraction of it, and you would need this SIP reduction factor as part of a calculation to determine the actual verification dose. Okay, VD max 15 substantiates a minimum sterilization dose of 15 kilogray. The maximum allowed was 1.5 adjusted for recovery, but this method is more restrictive than some of the other VD max methods. For very low bioburden, but the nice thing about it is you can use this for a very low bioburden and avoid using method 1, meaning you can test only 10 samples instead of 100 for sterility. It has its own separate verification dose table, and that one's table 10. The test results, acceptance criteria, whether you're doing method 1 or VD max, there will be a acceptance criteria for how many positives you can have on a sterility test. In method 1, if there's up to 2 positives out of the 100 you test, that is considered a passing test. On a VD max method, 1 positive is the limit out of the 10 tested. That at first is like counterintuitive. You're doing a sterility test, you expect to have no positives, but remember, you're testing to a probability. Your probability is that sterility assurance level. You're not going to release product at these doses. So if you meet the SAL, you've passed the test. In method 1, if it fails, meaning you get more than the 2 positives, and the outcome can be ascribed to an incorrect performance of a bioburden test, non-application of a recovery factor, maybe the dose was delivered incorrectly. Errors that can be corrected allow for the test to be repeated. Or if none of these apply, you would have to revalidate using another method. You wouldn't be able to just repeat method 1. With the VD max methods, you do have a little bit of a do-over. If you get 2 positives instead of the 1, you can repeat the test with 10 more samples at the same dose, but that second test, which is called a confirmatory test, can have 0 positives. If you think about it, the difference between 1 and 10 and 2 and 20 is the same thing. Okay. Method 2. We're not going to go into a lot of detail on this, but I want you to know it's there. It's not as widely used today because of how many samples it's needed, but the beauty of it is it cannot fail. It will always eventually reach a sterilization dose for you. It uses incremental dosing, or the resistance of the organisms on your bioburden, not the bioburden count, to determine the dose. You still need to know the bioburden and to have a technique for doing it, but the method 2 does not use dose tables. It does a series of calculations using data that you're going to develop through a number of sterility tests to set that verification dose and the minimum sterilization dose. To many, this is a method of last resort because it cannot fail. On very clean product, it also can give you the lowest possible sterilization dose, which is very attractive to products that have very low tolerance to dose. It tests the resistance of the natural population of product, so it can actually be a very low dose if you have only organisms that are easy to kill. Dose is not based on that total population data. It's based on the resistance. It has 2 versions based on how many incremental doses you do in the process of calculating your sterilization dose. If you look at the 2 forms of method 2, they use no bioburden samples to set the dose, but you have to have available sterility samples as high as 840 to do 2A, and 780 to do 2B. The difference is the increments of the doses you'll be testing. In 2A, you're doing it in 2 kilogram steps, so 2, 4, 6, 8, 10, work your way up. And 2B, 1, 2, 3, 4, 5, 6, 7, 8. With 2B, you cannot use a SIP. You would always have to test the entire product, but with 2A, you could potentially use a SIP. Okay, this is like my favorite slide. It shows you just how complex method 2 can be. What you're doing in method 2 is you're doing a series of incremental doses. You're looking at 3 different lots of product, just like you would do with bioburden in method 1. But now you're doing 20 samples per lot per incremental dose. And then you're testing all of those samples for sterility. There are 9 doses suggested for method 2A and 8 for 2B. One of the other contributing factors for why you need to have so many product units available is that when you get down to the verification dose, you will have to run 100 samples from a specific lot, one of the 3 you did the bioburden from, but up front you don't know which one that will be. So you have to have available all 300 samples. Method 1 at first seems kind of complex because you have to do so many samples, but when compared to method 2, it's actually a much simpler method. Okay. When can 2 be used? 2B, only when you have you can test the entire product, but 2B also has another limitation. If the radiation at any incremental dose, radiation at any incremental dose can have no more than 14 positives out of the 20. So you can't just have randomly everything growing all over the place. You have to have some naturally low amount of bioburden. And one of the calculated numbers, which is first no positive dose, cannot be greater than 5.5 kilogram. With method 2A, none of those restrictions occur. Okay. Why would somebody use method 2? Because you can get the lowest possible dose of any of the other methods if the product bioburden is low. Product that is very sensitive dose that can't be tolerated in any other form of sterilization, this may be your only way to do it. With the addition of table 6 in method 1, you'll see less and less of this method used, but it is still an effective way to get even a few more kilograms, which may mean whether or not you can sterilize a product with irradiation. Okay, can I use each of these? When can I use each of these methods? They all have dose tables that have limitations on them. The method 1 dose tables, actually as printed, go up to 1 million CFUs. They have sterility assurance possibilities from 10 to the minus 3 to 10 to the minus 6. 2A, 2B, you can do any number, but you're usually not going to bother with method 2 at all unless the dose has to be exceptionally low. Okay. Method 1 versus a VD max method, which is a more common comparison. The bioburden samples are the same. The sterility sample number is the one that's going to be different, but the sterility test is the same test. It just uses more of them for method 1. Okay. Dose audits. Once you've gone through all of this and determined what your minimum sterilization dose is, you early on determined what your maximum dose would be. You now have to maintain the product's claim over time. That's done with dose audits. So you have a test that you're going to do periodically, usually on a quarterly basis, to assure that the dose you're using still gives you the same sterility assurance level and that your product remains in process control as it's manufactured. You're going to do bioburden over, and you're going to do sterility testing over. Sterility will be done with the same number of samples you did initially in your validation at the same verification dose. This is your monitoring step. This is your biological data as part of monitoring your process. Okay. Each of the methods discussed validates that dose for 3 months. So if you make product every day for 3 months, you're doing 1 dose audit. You must do a dose audit quarterly if the product is produced to substantiate the effectiveness of the dose. Dose is not reset in an audit just because the bioburden may be slightly different, higher or lower. You do not change the verification dose. Use the same one. Bioburden and sterility testing, though, can be done concurrently, and they can be done while the product, the rest of the lot, is being irradiated for sterilization. The same pass-fail criteria apply for the audit as they do for validation. Method 1 and Method 2 audits are identical. So a Method 2 audit, you did a whole bunch of work up front to set the dose, but the audit is the same as a Method 1. You're doing the same 10 for bioburden as you did with VD Max, but you have the 100 for sterility testing. VD Max audits, 10 and 10. Dose audit frequencies, you're doing them quarterly, but there are the possibilities of reducing that frequency over time. After you have 4 passing quarterly dose audits, there is a possibility of reducing that to biannual audits, meaning you could do an audit every 6 months, then 3. This is not an automatically, you don't get out of jail free card. You have to have specific data on the bioburden, the types that you have in your population, not just pass 4 audits and you're done. Rarely, you also, I skipped a line, I apologize. After you go to biannual, you also have the possibility to go to annual audits. I personally don't know anybody who does annual audits because even biannual at least gives you data to account for seasonal changes, where an annual audit is an awfully long time to go by and not find out that you've had a change in your production process. Rules for reduction of the dose audit. Time is not the only criteria. You must pass all your audits. You must have data on the type as well as quantity of organisms. So bioburden in reality is not just a number. It is a makeup of a population of organisms that each have their own individual resistance. You must continue to do the bioburden, so you don't stop doing testing quarterly. You still have to do your quarterly bioburden. And the product's production process cannot change. If it changes, you're starting all over in your count of 4 passing dose audits. The risk is if you reduce the frequency, you've also reduced the data you have in response to any problem you might identify in the production process. But the advantages are clear. You'll be using up less samples, you'll have lower lab costs, lower radiation costs because you won't have to do that verification dose every 3 months. You might be able to do it every 6. And overall, you'll create a lot less paperwork. Note on the frequency of bioburden testing. Bioburden averages equal to or greater than 1.5 CFUs are required to be tested quarterly with your audits. Bioburden of less than 1.5 CFUs, where the sterilization dose is set with Method 2 or VD Max 25, also can do quarterly dose bioburden testing. But bioburden less than 1.5 CFUs, where the dose was set with VD Max 15 or Method 1, bioburden is monitored monthly, not just at audit time. And that's because 1.5 CFU is such a small number that even a minor change could have a huge impact on the sterility assurance level. If the outcome of the bioburden at this time, in this previous one, at the 1.5 CFU level exceeds the limit, you would have to do another audit at that time. So potentially, you run the risk of having to do a monthly audit rather than a quarterly. Okay. All we've done up to this point is we've cleared through the steps you need to get a validated dose range, a minimum and a maximum. There are a lot of individual things that may be specific to your product needs, so I suspect this is areas where you're going to have more questions based on the types of things you've seen. We're going to let you start answering questions now. We'll answer anything we can. And if you would prefer at some point to just discuss your product with one of our tech team members, by all means, feel free to contact us. That's what we're here for. We're here to help you through some of these questions you may have so that it makes it a little easier for you to decide what is best for your product. I thank you all for listening through all of this. There's a lot of information here, and that's why you've got a lot of things in tables so that you can go back and compare to see if you're really understanding the subtle differences of it. And the standards are there to help you. They're set up in different sections to make sure you can find the information you need. I thank you all for listening to me today, and we just leave you with one announcement that our next full live seminar will be in September.
Betty, we do have a few questions here. So,
Certainly.
and the first couple questions that came in are about the webinar itself. So I will let you take a super quick break while I answer those real quick.
Okay, thank you. I can breathe a little. Thank you.
Yeah. So the first one asked if there will be additional series to follow this one. Next week is part 3 and the final part in this series on the basics of gamma radiation. So that's the last one of this series. We do not have another series scheduled at this time. As soon as we do and we define those dates, we will make sure to let everyone know. As Betty mentioned, we do have a 2-day seminar and gamma facility tour coming up, the one that you see right there on your screen. If you are interested in learning about that, it's a really great comprehensive event. And you can find more information by visiting our website, steris-ast.com. And then the upper left-hand corner, you can click on the link for events to find all about that. And that's where future webinars will be listed as well. The next question was, can we get a copy of all the PowerPoints in this series? We do not provide copies of the PowerPoint slides, but we do record all of the sessions. Today's session was recorded as well. And shortly after the webinar ends, we will download the recording, we'll put it on YouTube, and then everyone that registered will get a copy or a link to view that recorded webinar on YouTube so you can view it at any time. So those are my questions, Betty. I'll start handing over the technical questions to you. And the first one is, which standard outlines the procedure for validation of method for bioburden?
11737-1.
All right, and the next one is, if 2 components are packaged separate but are used together in procedure, do you add the bioburden together to establish the verification dose? And are there any cases when the bioburden can be treated separately?
You can actually do it either way. I see some people that will validate them as 2 separate products for one really good reason. Should there be a problem with one, it doesn't affect both of them. But to simplify the validation, many times they are tested together. But that means if there's a problem with either one, both of them fail. So you can add them together to test them, or you can test them as 2 separate products and 2 separate validations.
Okay, thanks. Another question that came in regarding the webinar is, if we have questions after this webinar, who can we speak to? And we will send contact information in the follow-up email right after this webinar for Betty and her team on the radiation tech team.
Mm-hmm. Yeah, we look forward to that. You can ask anything you want and be as interactive as you want in case you want to discuss something about your product. We're open to that at any time.
All right, the next question is, can you provide the reference for only 3 organisms needed for medical device BNF testing?
Yes, it's in USP. It is mentioned in 11737-2, but it's one of the USPs. Email me that question, Sarah. I want to look at the specific number for it, but it is in the USP.
All right, next one is, regarding the positive sterility test after verification dose, are there any requirements to identify the organism species?
Technically, no, but I would highly recommend that you do, because if you get a sterility positive, that is your weakest link. That is the thing most likely to create a problem later. It may be something that is easily identified and you can tell where it came from, and you can eliminate it from your production process. So it makes your production process more robust to know. Plus, the more information you have on the types of organisms present in your product over time gives you data to support any problems later, but also gives you some of that data you need if you ever want to reduce your audit frequency.
All right, on table 9 for VD Max 25, starting at 120 CFUs per item, the verification dose starts going down instead of up. And they're asking, can you explain the reasoning for this better than the ISO does?
The basic thing, the best way to look at that is, when you get to 25 and 1,000 CFUs, take a look at the method 1 table for 1,000 CFUs, and you will find that that number actually is the same. they have to give you the ability of those two tables to converge, so the mathematics of that slope starts to come back down. And if you think about it, you're getting a very generous dose if your bioburden is very, very low with a VD max. But by the time you start getting closer and closer to the cutoff, you have to match what's in method 1, and you can't get more dose than you would get to method 1.
All right, next question. What would be the best course of action if the quarterly bioburden for VD max 25 exceeds 1,000 CFUs?
Okay. The question would be first, is it one sample or is it a whole bunch of samples? I would look, if I had outliers, I would look at the identity of what was in that bioburden to see if maybe it's something obvious, like maybe it was a handling issue, maybe there was a torn glove when somebody was working with it. Most people will take several different possibilities. First thing I would do is ID it. Because you might find that, for example, it's got something on it that should have been like a staph that should have been very easily killed by whatever your verification dose was. If it was to show up after it's been irradiated, it's probably a handling issue. If it's something that was very difficult to kill, that may be something that wasn't present before, so it can give you some useful information. I see different people in their SLPs have different processes put in place for how they will handle that. Usually if it's one sample, it's not as much of a concern, but if it's multiple or it seems to be a trend, you may, for example, repeat a bioburden test with some more samples to get more data to see if it's consistent or if it was a one-off outlier. There may not be an exact answer for that. A lot of times it's going to be what you put in place to monitor your process.
All right, next one. Oh, and Betty, somebody did clarify the USP number. It's USP 71, so thank you for that.
I don't know that the BNF is in there. That's the sterility one. That's why I want to check. I think it's 30 something, but I want to double check. But thank you. We'll double check both of them to make sure.
Okay, next question. Is there a specific reference in the ISO to not change dose in between dose audits, even if you get changing bioburden data?
Yes, it's in there. It says to use the original verification dose, because what you're doing is you're monitoring production process control. And remember, you are not testing every lot you make. You're going to have natural fluctuations in your bioburden, but they should all be within the response of that dose in order to assure that lot to lot, since you didn't test as well as the ones you did, that your sterility assurance level can still be met.
Right. Is the validation process and testing for the E-beam irradiation the same as gamma?
Absolutely the same.
All right. Is there an acceptable minimum allowed percentage of CFUs recovered in inoculated product recovery test?
There's not a specific, but there is a rule of thumb. If you have a recovery efficiency of less than 50%, it is recommended that you repeat the test or use a different test method. Maybe improve the method, or for example, if you were doing exhaustive rinse, maybe go to inoculated. The reason for that is, think about what's happening. If you have a 50% recovery, you are automatically going to double your bioburden number. That's a huge increase. It also means that you're not getting a very accurate idea of just how many bioburden units you have. So to try to improve it is to your advantage. In most cases, if it's over 50%, most people are not going to repeat it. But you want to be aware of that number, because a lab, when they see a number that low, they will come to you and say, do you want us to repeat it? Because what happens if it's only like 15%? That's a huge correction factor, more than double. And you could be coming up with numbers that don't really make sense. But bottom line, it means you're not accurately representing your bioburden, and the bioburden is what's driving your sterilization dose.
All right, next question for VD Max 25. If we have an average bioburden of 1,000, would our minimum be 8.1 and max 25 kilogray?
No, your minimum becomes 25 kilogray, and your verification dose becomes 8.1. I'm going to assume that's where the 8.1 number came from, because I don't have the table in front of me. But you never will have a minimum dose lower than 25 kilogray if you use VD Max 25.
Okay, and we just have a few more questions here. Could you provide the clause in the standard in which we can review the information related to the percentage of recovery for the bioburden process?
I don't know the clause, but it's in 11737-1. It says in there that it is recommended if the if the recovery is low, to repeat it. There are products that will have naturally low recovery efficiencies even if you repeat them, but usually you're going to be expected to have at least repeated the test a second time to see if you can improve it.
Okay. Is there any specific value for the percentage of recovery for the bioburden process where we can review this value? Or where can we review this value?
I don't understand the question. Repeat it, Sarah. Maybe I missed something.
It says, is there any specific value for the percentage of recovery for the bioburden process? Where can we review this value?
Okay, the value of the recovery efficiency is to assure the accuracy of the bioburden number and directly to assure that the sterilization dose and verification dose are accurate. Most labs will recommend that you periodically repeat that test to assure that there aren't changes in your product, which that could be the question. If I'm missing it, whoever put in the question, just email me so that we can talk it through. But I think those are the two areas that would probably relate to that question.
Right. Can you use biological indicators for product sterility testing? If so, are you allowed positives?
Well, in the standards for radiation, it is said that it is not recommended to use biological indicators for radiation. The main reason is that they don't accurately represent they're one organism with a not exceptionally high d-value, or the dose at which they would be dropping a log in population. They are also biological, so they can be variable. You can use them, of course, they are your choice, but if you're validated to ISO, you are going to be expected to pass sterility at that verification dose. You would have in your procedures, if you had the BIs, my guess is your procedure would say that all the BIs have to be killed. So if you have that in your procedure and one was to survive, even if it was one that should not have logically survived, like the one on the outside of the box, but the one in the very center was killed, you are going to be faced with a dilemma. You are probably going to have to reject that product. So I wouldn't recommend using them because they don't give you any value that the validation process and verification dose don't already give you. And you're also using, in radiation, dosimetric release. So you're always measuring each batch you release to assure that it's gotten the appropriate dose to claim sterility.
All right, we had a couple more come in. Right now we have 4 more questions in the queue here. We'll answer these 4 questions and then we'll wrap it up. And then just to let anyone know too, I did see that I had a few questions come in to me via email, which we'll send to Betty and she can respond to you too. So if you did send an email, those will be addressed. The next question here in our panel is, can you speak to the use of adverse substance screening in lieu of BF testing?
I don't believe it's in lieu of. Adverse substance screening is more related to bioburden, to assure that you're not you're getting accurate numbers with the bioburden test. BNF is directly related to the sterility test conditions.
How is bacterial endotoxin or LAL testing with every lot you produce different from the quarterly dose audits?
Okay. LAL is not related to dose setting or to your sterility assurance level. It's to monitor the presence of a particular type of protein that comes from certain types of organisms. It's related to the cause of a pyrogenic effect in humans. So if your product is labeled, for example, pyrogen-free, or is used in a situation where that type of organism present could be severely damaging to a patient, you are required to test every lot you make for endotoxin. You can test the endotoxin on the sterilized products. So for example, if you sterilize 25 to 40, you can take samples out of your production run and test them for endotoxin. The reason you test every lot is one of the largest contributing causes to the presence of an endotoxin in a product is a water supply. And water supplies can be tremendously variable within a day, so that things you made early in the day could be different than ones later in the day, so you take random samples. It's a limited number of samples you're required to test based on the batch size, so it would never be more than 10 samples, and it could be as low as 3. But it is not related to the dose audit at all. It is a separate test unrelated to dose setting.
Okay, is it possible to augment a dose established with VD max 25 when the average bioburden is greater than 1,000 CFUs, or would another method need to be used?
Okay, augmentation is done when the sterility test fails, not when one sample has been was higher than 1,000 CFUs. You, of course, can always augment your dose. That is your choice, as long as your dose is over the amount needed to make your sterile claim based on your validation. So augmentation is, for example, say you got 2 positives on your sterility test when you were only allowed 1. Or better, since you can do you can do a confirmatory test, let's say you got 3. There are calculations for determining how much more dose you would have to give to that production batch to assure that the sterility assurance level was still met while you're investigating the cause of the increased bioburden.
All right, and last question. If your minimum is 25 kilogray, how do you develop the max then? Just keep testing at higher dosages to see if your product can withstand the dosage, or is there something else that needs to be performed?
Okay, it is on some level trial and error, but the critical bit will be how much dose the plant that's processing your product needs in order to process it. Rule of thumb, most people are going to start at about 1.6, so that's where the 25 to 40 comes from. You can test multiple doses to see if you're not required to take it to failure, but sometimes you want to know a dose at least a bit higher than you plan to use because of the fact, and whoever asked the previous question, perfect timing, because there might be times when you need to augment your dose. If you've ran product 25 to 40 and used most of that dose range, you may not have any dose left in that range to augment, but say you tested to 50, but you only run 25 to 40, you got that extra 10 kilograys of reserve.
Okay, thank you, Betty, and thanks to everyone for joining us again today. Our Gamma webinar series concludes next week with Part 3, Gamma Radiation Dose Mapping. If you haven't already done so, please register separately for next week's presentation. We will send a link to register in the follow-up email from today's webinar that you'll receive later today. And the email will also include a link to view the recording of today's webinar on YouTube. Thanks again for joining us today, and we look forward to learning with everyone again next week. This concludes today's webinar.